Hey y'all. So today we'll be going over Chapter 2, which is notoriously one of the harder chapters of the first exam, and that's mostly just because it's largely kind of biological, which can be challenging for some students. So just be aware that you probably want to devote a little extra attention to this chapter when you're studying for the exam. Make sure you have out your pre-session worksheet and that you're filling it out as we go, and we'll go ahead and get started. So phrenology was this pseudoscience, and what a pseudoscience means is it was trying to be a science, but it really wasn't scientifically valid, so it failed to do anything really helpful in the scientific community. So phrenology was a pseudoscience where a researcher would touch a person's head and try to feel what parts of the skull were more bumpy than other parts, and those bumps on the skull supposedly correlated with certain traits. So if you had a really big bump maybe over your ear, that might mean that you have a really great sense of humor, right? But if that bump over your ear is very small on you compared to other people, then you have no sense of humor, that sort of thing. Or, in another place on your head, it could tell intelligence, or it could tell extroversion, for example. But of course, this was not actually true. This was a pseudoscience, but that's what phrenologists did believe at the time. And so the reason that it was debunked was because it was incorrect, but mostly because different phrenologists would give different interpretations to the same people, and so the practice wasn't scientific. It was inconsistent, it was unreliable. And Mark Twain specifically told of a time when he went to a phrenologist who said the part of his skull that corresponded to his sense of humor was really small, so he was without a sense of humor. But he went to a different phrenologist who then told him that he had the largest bump for humor that they'd ever seen. So if you went to 2 different or 3 or more different types of phrenologists, you could get inconsistent readings with them, which is what invalidated it as a science. But on the other hand, there was one thing that phrenology had right, and that's what we call the localization of function. And that is just the idea that different parts of the brain are used for different things that you do, rather than your entire brain being engaged in every single activity. So you have a part of the brain that functions for memory. You have a part of the brain that functions for, practical intelligence, for example. There are different parts of the brain that do different things, as opposed to the whole brain being used. So the localization of function was what phrenology did have right. And then biological psychology just studies the link between biological and psychological processes, so between what's going on inside your body, your hormones, and all those sorts of things, and then what's going on inside of your mind. Okay. So we'll go through this somewhat quickly, and I actually have a picture on the next page that might be a little more helpful. But basically, you just want to know all of the different parts of a neuron. And so the dendrites are these little bushy extensions that are on the end of a neuron. And the dendrites just bring in incoming information. So when an electric impulse is generated, the dendrites are what receives that electrical impulse and then passes it along the rest of the way of the neuron. And so the dendrites are these bushy extensions at the end, and then we have the cell body. And just know really that the nucleus lies within the cell body. And then from there, the axon is just the longest part of the neuron. And sometimes, on some neurons, we have myelin sheaths. Myelin sheaths are just encasings that go along the axon, and their purpose, you'll want to write this down, the purpose of the myelin sheath is just to speed up the impulse, so it makes it travel faster across the neuron. So only some neurons have the myelin sheaths, just the ones that need to function more quickly than others. So the neurons inside of your brain and inside of your fingertips and stuff like that are going to be myelinated, or going to have myelin sheaths, whereas the neurons, let's say in your kneecap, for example, wouldn't, because you don't need to receive information from your kneecap as quickly as you need to receive information from your brain or, from touch, when you touch something with your hand, your fingertips. And then at the end, much like at the start with the dendrites, we've got these axon terminals, which is just the last part of the neuron. So really what you want to know here is the order of these. Know that it starts with the dendrites, then moves to the cell body, then across the axon, which may or may not have myelin sheath, and then the axon terminals is the last step in that process. I'm going to go back really quick because we need to talk about glial cells. And so this is the first thing that I want you to write down on your worksheet that I should see highlighted. This is one of those traps that I mentioned. So next to glial cells, I want you to write that glial cells are the parents of neurons. And the reason that I say that is because glial cells function to clean up after neurons and care for them. They provide them with nourishment and protection, and they also just kind of clean up the junk around them, like the extra neurotransmitter and stuff like that. So glial cells are like parents of neurons. Okay. All right, so this is probably one of the more confusing parts for people, is understanding how these neural impulses work. I'm going to try to break it down as much as I can and make it as straightforward as possible. But an action potential, really simply put, is just an electrical charge. And sometimes you have an action potential that's generated, but the neuron doesn't fire. And sometimes you have an action potential generated that's strong enough to make the neuron fire. But kind of the difference between an action potential that does make the neuron fire and one that doesn't is whether or not it reaches the threshold. And the threshold is just the minimum level of stimulation that you need in order to trigger a neural impulse. And so the analogy that we often use in the textbook is like firing a gun. So just because I push the trigger a little bit doesn't mean I'm going to be able to make the gun fire. There's a certain amount of minimum level of intensity that I have to push the trigger hard enough to actually make the gun fire. And that's the same thing with neurons. You have to reach that threshold, that minimum intensity. I have to push the trigger hard enough to actually get it to fire. And that's what we're looking at with neurons. So if the action potential is strong enough that it reaches the threshold, so you can think of it like if the action potential is me pressing on the trigger. If I press hard enough on the trigger that it reaches the threshold, the gun will fire. If the action potential is strong enough to reach the threshold, then the neuron will fire. But if the action potential is not strong enough to reach the threshold, then the neuron will not fire. So the threshold is that level of stimulation that you need in order to actually get the neuron to fire. The refractory period is this idea that once a neuron has fired, it can't immediately be stimulated to fire again. There's a small waiting period where it kind of has to reset itself, and that period is the refractory period. The purpose of the refractory period is just to stop neural impulses from actually traveling backwards, but that's a little more complex than what you need to know for the exam. So just know it's a rest period where the neuron can't fire again for a certain amount of time. And then you can generate another action potential, and if it reaches the threshold, then you can fire again. And then the all or none response. So this is the idea that a neuron is either going to fire at full power or not fire at all. It's not possible to have a neuron fire like at half speed or something like that. So whether, the action potential is just strong enough to reach the threshold or way stronger than it needs to be to reach the threshold, that's not going to make the neuron fire any faster or any harder. The same way with firing a gun. If you push the trigger harder, it doesn't make the bullet travel faster. But if you don't press the trigger hard enough, then the bullet doesn't fire at all, right? Another analogy that I like to use is flushing a toilet. And so the action potential would be pushing down the lever on the toilet, and if you push it down hard enough and it reaches threshold, then it makes the toilet flush, makes the neuron fire, makes the toilet flush. But if you don't push it hard enough, then the toilet just won't flush at all. If I really slam my hand down on the lever, it's not going to make the toilet flush harder or faster. That's the idea. It either flushes or it doesn't. The gun either fires or it doesn't. The neuron either fires or it doesn't. doesn't fire at half speed or double speed or anything like that, double intensity, none of that. Okay. So when it comes to neurotransmitters, this is going to be largely kind of where you've just got to commit some stuff to memory. But know that neurotransmitters are chemical messengers of the nervous system. They travel from sending neurons and then attach or bind to receptor sites on the receiving neurons. So from the sending neuron, it would leave the axon terminal and then bind to the receptor sites on the dendrites of the new neuron. Okay, so the most important three here are going to be the first three. You want to really commit acetylcholine, dopamine, and serotonin to memory, and also what not enough or too much of them causes. So know that acetylcholine is related to memory, and therefore, if you don't have enough acetylcholine, you can end up with Alzheimer's disease. So luckily that starts with an A, and so does Alzheimer's disease, so that's how you can remember it. Acetylcholine and Alzheimer's go together. Acetylcholine helps with memory, so if you have not enough of acetylcholine, then you have memory loss, Alzheimer's. Next is dopamine, which has to do mostly with emotion. That's probably the most important one, but also movement is largely important too. And so if you don't have enough dopamine, you can get Parkinson's disease. You definitely want to have all four of these things memorized: the Alzheimer's, Parkinson's, schizophrenia, and depression. So not enough dopamine, Parkinson's disease, but too much dopamine leads to schizophrenia. And if you know anything about schizophrenia, we'll get there in chapter 15, but you probably do know that schizophrenics experience delusions or hallucinations a lot of the time. And what that is related to is just too much dopamine activity going on in the brain, that their brain actually uses all that extra activity and turns it into a perception that isn't really there. So it turns it into a hallucination, seeing something that isn't really there. It's misinterpreting the overactivity of the dopamine inside the brain. So that's how you can remember the dopamine, too much of it, is related to schizophrenia. If there's too much, then the brain misinterprets it and creates false experiences. And then serotonin is largely related to mood and arousal. Those are probably the most important ones, mood and arousal. And if there's not enough serotonin, you end up with depression. The biggest mistake that students make on this portion of the exam is that they want to say that dopamine goes with depression and serotonin goes with schizophrenia because they want to match the D's and the S's. Don't make that mistake. Know that it's switched. Dopamine goes with schizophrenia, too much dopamine, and serotonin goes with depression, not enough serotonin, because lots of serotonin's kind of the happy neurotransmitter, right? So if you have a lot of it, your mood is really elevated. But if you are without serotonin or it's depleted, now we have an issue with depression. And then I would, to some degree, know these last four, but they're not nearly as important as the first three. So when it comes to norepinephrine, just know that that has to do with arousal. And maybe that last part of epinephrine can help you to remember that. And not enough norepinephrine leads to a depressed mood, which is different than depression in the fact that a depressed mood is a temporary emotional state, whereas depression is a long-term clinical diagnosis. And then what you do want to know about GABA and glutamate, and this is really all you need to know about GABA and glutamate, is that GABA is an inhibitory neurotransmitter. Glutamate is an excitatory neurotransmitter. So I like to think of GABA as being really slow and inhibited and just kind of, boring, right? And then glutamate just bounces off the walls. Glutamate is so excited all the time, right? So that's how you can remember those. But just know that GABA is inhibitory, glutamate is excitatory. That's often a test question. And then endorphins, just know really that those influence the perception of pain. I really cross that out. Perception of pain and pleasure. Okay. And then agonists versus antagonists. So an agonist will actually help to increase the effect of a neurotransmitter, whereas an antagonist decreases the action of a neurotransmitter. The way that I remember that is if something is antagonizing you, you'll probably stop doing whatever you're doing, right? Because we don't want to be antagonized or attacked by someone. So if something is antagonizing you, if we have an antagonist, it's going to make you stop doing whatever you're doing. So it's going to decrease the action of the neurotransmitter, whereas an agonist will help the neurotransmitter to create its effect. Okay. So go ahead and think about this. Maybe pause the video and try to think about this a little bit. But if you're trying to treat depression, would you want a medication that behaves as an agonist or an antagonist? Well, if we know that depression is linked to an undersupply of serotonin, then we would want an agonist to increase the firing of serotonin. If a patient had migraines brought on by an overconsumption of MSG, will we want an agonist or an antagonist? Well, it's an overconsumption, meaning too much glutamate. MSG stands for monosodium glutamate. And so we would want an antagonist to reduce the firing of any additional glutamate in the patient's brain in order to stop the migraines. So these are really good application questions where you kind of have to know two parts of it. You have to know, what neurotransmitter you're dealing with and how it relates to what we're talking about, depression or migraines. And then you're going to need to know what an agonist or antagonist does, right? So that you can put these two pieces together and figure out how to answer this type of question. Alrighty. This is really important. These next three questions make such easy test questions that get people really confused. So be clear about what's going on here. Know that the central nervous system is just your brain and spinal cord. That's it, just the brain and spinal cord. And then your peripheral nervous system is everything else that's part of your nervous system besides the brain and spinal cord. So it's the sensory and motor neurons that connect your brain and spinal cord, your central nervous system, to the rest of the body. And then there are subdivisions of the peripheral nervous system, and those subdivisions are the autonomic nervous system and the somatic nervous system. And then the autonomic nervous system is further broken down into the sympathetic and parasympathetic nervous systems. I have a little graphic on the next slide that will help clear that up a little bit too. But know that the peripheral nervous system has two subdivisions. The first one we'll talk about is the somatic nervous system, and that is your voluntary movement system. So it controls your body's skeletal muscles. So really what the somatic nervous system does is it enables you to do any kind of physical action that you make the conscious choice to do. So to pick up a pencil or to take a drink of your water. All of these things, to talk, to chew food, all of these things are somatic nervous system activities because they are voluntary muscular actions. Whereas the autonomic nervous system is automatic. I want you to write that down. It's another one of those traps. The autonomic nervous system, think of that as the automatic nervous system. So that is for involuntary movements, such as, your heart beating or your lungs breathing. You don't have to tell your heart to beat. You don't have to tell your lungs to breathe, that sort of thing. So automatic movements of the body belong to the autonomic nervous system. And so largely the autonomic nervous system functions with internal organs. And then, like I said, there are two subdivisions of the autonomic nervous system. The first is the sympathetic nervous system, and then the parasympathetic nervous system. And these two systems are related mostly to like fight or flight type responses. So the sympathetic nervous system arouses your body for fight or flight. So it gets you ready to deal with any kind of incoming threat that you might have. Whereas the parasympathetic nervous system, I think parasympathetic, paralyzing. Parasympathetic, paralyzing. It calms your body down after the threat has passed. So here's that little graphic. Got the CNS, which is just the brain and spinal cord, PNS, peripheral nervous system, that is divided into the somatic nervous system, voluntary movements, and the autonomic nervous system for automatic functions. And then the sympathetic nervous system arouses the body for fight or flight response. It increases your heart rate, increases your respiration, all those sorts of things. And then the parasympathetic nervous system, parasympathetic, paralyzing, calms your body after that threat has passed. So it's going to decrease your heart rate, decrease your respiration, etc. But the one thing that is sort of opposite for both of these is your digestion. So when it comes to the sympathetic nervous system, this actually decreases digestion. But your parasympathetic nervous system increases digestion. And this is really silly, but the the reason for this, honestly, is that if you are in a fight or flight situation, so your sympathetic nervous system is activated, it's not going to be helpful for you in a fight or flight situation to all of a sudden need to go to the bathroom. And so that's why your sympathetic nervous system decreases the rate of your digestion so that you don't need to go to the restroom right away. But once your parasympathetic nervous system is kicked on, the threat has passed, you're relaxing now, that's your rest and digest system. So that will increase digestion now. Everything else should be in the opposite direction. Like the sympathetic nervous system should increase most things, your heart rate, your respiration, all that. Function of your sweat glands too, stuff like that. Whereas the parasympathetic nervous system generally decreases those things. The only thing that works in the opposite direction is digestion, and that frequently does come up on exams. So do make sure that you are aware of that. When it comes to the endocrine system, really what you need to know here is we've got chemical messengers in both the nervous system and the endocrine system. And I want you to write this down, that the chemical messengers of the nervous system are neurons, whereas the chemical messengers of the endocrine system are hormones. Hormones. The endocrine system is all about hormones in your bloodstream. And so hormones, like I said, are chemical messengers that travel through the bloodstream and affect tissues of the body and brain. They influence a whole variety of things like hunger, sexual arousal, aggression, other things too. But yes, chemical messengers of the nervous system are the neurons, and the chemical messengers of the endocrine system are hormones. Sometimes on test questions, they'll talk about chemical messengers of one system versus the other, and some of the answers might be mixed together, where some of the answers are hormones, some of the answers are, neurons. And so you need to know which one goes with which system so that you can immediately cross out some wrong answers in those types of questions. And then the adrenal glands, just know that those lie above the kidneys and they secrete hormones like epinephrine and norepinephrine. Think adrenal adrenaline, right? And so epinephrine and norepinephrine are those hormones that function when your adrenaline is high, right? When you need to be really aroused or have a lot of energy. And then your pituitary gland, know that we call this the master gland. It's sort of it controls all of the other glands of the body. So the pituitary gland is really important. It's the master gland. It's the powerhouse, the control center of all the other glands in the endocrine system. And its controller is the hypothalamus. This is the most influential gland of the system, and it regulates the body's growth and controls all the other endocrine glands. Okay, so here we've got all these different types of tools of neuroscience. And These are important to know the difference between because these make really good multiple choice questions. They'll just give you any 4 of these 5 as multiple choice answers, and you're going to need to know specific ways to tell these apart from each other. So I want you to write down 1 or 2 words about each of these as I go. This is another thing I'm going to be looking for on your worksheet. So next to EEG, electroencephalogram, write down the word electrodes. Electrodes. The EEG is all for recording waves of electrical activity across the surface of your brain. So this is a non-invasive procedure. This is done by electrodes being hooked up to your scalp. So nothing actually goes inside of your head or brain or anywhere in your body. This is all on the surface of your skin. And then next to magnetoencephalography, MEG, write down magnetic fields. Magnetic fields. This is just a brain imaging technique that measures your electrical activity with magnetic fields. So the EEG measures electrical activity with electrodes. The MEG measures electrical activity with magnetic fields. So that's also a non-invasive procedure as well. The PET scan is all about glucose. Glucose, glucose, glucose. If glucose is mentioned in the question stem of your multiple choice, guarantee you the answer is PET scan. It's all about glucose. And so that is just tracking a radioactive form of glucose that moves while your brain is performing a task, so we can see what part of the brain is active while you're doing any given task. And then here's how I want you to think about MRI and fMRI. Think of MRI as a single photograph of your brain, whereas an fMRI, think of it as a burst of photos. So when you go to like your camera app on your iPhone and you hold down the picture taking button and it takes like several photos all in one burst, that's basically what an fMRI does. So an MRI takes one single photo. An fMRI takes a bunch of successive photos, one right after the other, so you can see small movements in the brain. It's kind of like a flip book almost. So write down single photo next to MRI and photo burst next to fMRI. I have a couple photos here. So yes, EEG, like I said, has to do with electrodes being placed on the brain to measure electrical activity. MEG is all about those magnetic fields being used to measure electrical activity. PET, you're looking for glucose that's moving while the brain's working. MRI is a photo that shows just anatomy of the brain, whereas an fMRI, successive photos, can also show you function and structure beyond just anatomy. Okay. So older brain structures. The brainstem is at the beginning of the spinal cord, and it's responsible for your automatic survival functions. So it's the oldest part, central core of the brain. Automatic survival functions. The medulla is at the base of the brainstem, and it controls heartbeat and breathing. I don't know why, but the medulla seems to come up more on exams than all of these other ones. So just really be aware that the medulla is heartbeat and breathing. Thalamus is on top of the brainstem, and it is the sensory control center in your brain. So whenever you touch something and the sensation that you feel on your fingertips, the reason that you can feel that sensation is because of the processing work that your thalamus does to interpret that sensation and tell you sort of what it feels like, to give you the sensation that you get. The reticular formation, know that that is associated with arousal. For some reason, that comes up a lot too, that the reticular formation is all about arousal. So if you're feeling particularly drowsy, you feel like tired, you want to go to sleep, there's something going on with your reticular formation. It is not as active as it should be to sort of arouse you. And that travels through the brainstem and into the thalamus. And then the cerebellum, we also call the little brain. It sits at the rear of the brainstem, and it does all sorts of things, but mostly processes sensory input and coordinates your motor output. So it takes in the sensations that you feel and then coordinates your output to that. So, you know, when you touch something and it's hot, it's your cerebellum that takes in the information that it's hot and then tells your body pull away from it. That's your motor output. It tells your body to pull away from it. And this also enables nonverbal learning and memory. When we get to the memory chapter, we'll talk about how, you know, it's responsible for implicit memories and, you know, the hippocampus is for explicit memories, but we'll get there. At this point, just know that it's associated with learning and memory, but you don't need to know the details yet. Oh, and then what these older brain structures have in common, and you should write this down, is that they are all for survival functions. All for survival functions. All of these older brain structures deal with survival. And that's really important because what it tells us about how our brain processes information and how our brain develops is that the most important parts that just enable us to survive developed first. Notice that this is the slide for older brain structures. So these were the very first brain structures to ever develop. And then as time went on, you know, more adaptations were created to help us cope with the environment in which we live. But at the base of everything is just this idea that we needed to be able to survive. And it's sort of evolutionary psych that studied and came up with all this. But just know that the oldest brain structures enabled survival functions. So our brain starts with the most important, most fundamental survival instincts, and then we move up to sort of higher level thinking and processing after that. The limbic system. This makes also fantastic, easy test question because there's exactly 3 structures within the limbic system. And so it's really easy to ask you things like, you know, which of the following is not a structure of the limbic system? But you want to know what the 3 structures are and what they do. So the first structure in the limbic system is the amygdala. That is for fear and aggression. That will come up over and over and over and over again. So know about the amygdala for fear and aggression. The hypothalamus lies below the thalamus, the stem hypo means below. I think that's like a Latin derivation. But it directs maintenance activities like eating, drinking, and body temperature. And it does help govern the endocrine system, but really, this is what you want to know. That the hypothalamus regulates the systems of your body. So it is sort of functions in telling you whether you're hungry or thirsty, telling you if you're too hot or too cold. It's just sort of a maintenance system for your body. And then the hippocampus is the third one. Now, be aware that a lot of people mix it up and they think, oh, if the hypothalamus is part of the limbic system, then the thalamus must be too. But that is not the case. It's the amygdala, the hypothalamus, and the hippocampus is the last one. People almost always forget hippocampus and try to replace it with thalamus. So don't make that mistake. The hippocampus is the last one. And the hippocampus processes memories. The way that I remember that is if you saw a hippo on campus, you would probably remember it, right? So the hippocampus is all for processing memories. Now, specifically explicit memories, but again, you don't need to know about that until we get to chapter 8 and we actually talk about the difference between implicit and explicit memory. So for now, just know that the hippocampus is your memory storehouse. Okay. So the cerebral cortex and the lobes of the brain, what you're going to want to know is what cortex lies within each lobe. So the cerebral cortex, that's not one of the cortices I'm about to talk about, but the cerebral cortex is basically the whole cover over your brain. So the cerebral cortex makes up most of your brain. I think it's like 65% of your brain is cerebral cortex. but it's just the cover over your entire brain, and it controls the body and processes information. It's sort of the first thing that any sensation touches, and then the last thing that it touches as things leave your brain and travel to the rest of your body. And then there are things called association areas along the cerebral cortex. You don't really need to know that much about them, except that they are involved in higher mental functions like learning, remembering, thinking, and speaking. So then, these are the 4 lobes and corresponding cortices that you're going to need to know. The frontal lobes are just behind your forehead, and those are involved in speaking, muscle movements, planning, and judgment. You've got the motor cortex within the frontal lobes, and that controls your voluntary movements. So that's a somatic nervous system function. In your parietal lobes are all about sensation. We're receiving sensory input for touch and body position. And the cortex in there is the somatosensory cortex, right? So if you know that the parietal lobes are all about sensation and sensory input, then you should be able to remember that the somatosensory cortex is what's inside of the parietal lobes. And the parietal lobes are sort of in the middle of the head behind the frontal lobes. Then you've got your occipital lobes. Those are for visual information. So, surprise, surprise, they have the visual cortex inside of them. Visual cortex. And then the temporal lobes are right above your ears. Those are for auditory information. So again, surprise, surprise, the cortex inside of your temporal lobes is the auditory cortex. So with occipital lobes, you have the visual cortex. Temporal lobes have the auditory cortex. Also, fun fact, your occipital lobes are at the back of your head, right? So all the way in the back, kind of by your brainstem. And so if you ever need to knock someone out, you want to hit them in the back of the head, because that is where their visual cortex is. So if you need to make someone, see stars so that you have time to run away or something like that, you want to hit them in the back of the head in that occipital lobe, in the visual cortex, because it will black out their vision and make them see stars. Okay, and then lastly, the corpus callosum. I can promise you that you will have a question on the corpus callosum on this exam. There is always at least one. So the corpus callosum connects the 2 hemispheres of your brain. It's just fibers that exist between the left and right hemisphere and connect it together. And so sometimes we have something called split brain, which is where there's something going on where we have to cut those fibers between the left and right hemisphere of the brain. We have to sever them, cut them. And when that happens, it's sort of like you have 2 distinct brains, both inside of your head. And so we have a condition called split brain, where the patient's 2 brain hemispheres no longer communicate with one another. And so usually split brain patients act pretty normal, but under laboratory conditions, we can see sort of really interesting differences between a split brain person and someone whose corpus callosum has not been severed. I think the best way for me to show this to you is to draw it out, which I have done. Sorry, I am not an artist. But what you need to understand is that there are 2 crossovers that happen. And so the question for the corpus callosum problem will say something like, a picture of a mouse is flashed in a split brain patient's right visual field, and a picture of a cat is flashed into their left visual field. And then it will ask you one of 3 questions. First question is, which of the 2 images, mouse or cat, will the patient point to with their left hand? Second question is, which of the 2 will the patient point to with their right hand? And the third possible question is, which of the 2 will the patient say that they saw out loud? So what you need to understand is that anything flashed to the right visual field is communicated to the left hemisphere of the brain. The right visual field connects to the left hemisphere of the brain. And the left side of the brain controls the movements of the right side of the body. You may have heard that before if you've ever taken like an anatomy class or physiology or something like that. And then same way, the other way, the left visual field communicates to the right side of the brain, and the right side of the brain controls the left side of the body. So when we're looking at, let's use that example of a mouse being flashed into the right visual field and a cat being flashed into the left visual field of the patient. Well, that image of the cat is going to be communicated to the right side of the brain, whereas the image of the mouse is going to be communicated to the left side of the brain. So because the left side of the brain controls the right side of the body, the patient will point to the mouse with their right hand, and the patient will point to the cat with their left hand. See how that works? So if you kind of draw yourself a little chart, even if you don't, make it as complex as this with like drawings or anything, but just, visual field, brain, and body. And then you just fill it in, mouse, cat, and then alternate. Cat goes over here and over here. Mouse goes over here and over here. This will be substantially helpful to you in figuring out how to answer that question. So that answers 2 of the questions. What do they point to with their left hand? Cat, because that was in the left visual field, got communicated to the right brain hemisphere. The right side of the brain controls the left side of the body, so the left side of the body points to cat, and then vice versa for the right side of the body pointing to mouse. Now the third question that they can ask you is, which of the 2 will the patient say that they saw? And what you need to know is that it's only the left side of the brain that does any kind of speaking. So the left side of the brain is reserved for speech. Speech, speech, speech. So when they ask you, what will the patient say that they saw? It's whatever is inside their left brain hemisphere. So they will say that they saw a mouse. And that will always be the same thing. Whatever they say they saw will also be the same thing as what they point to with the right side of their body, because the left brain speaks and the left brain controls the right side of the body. Okay? So let's look at an example question. So if a split brain patient sees the word he in the left visual field and the word art in the right visual field, when asked to say which word he saw, the patient will say what? Okay, so we've got left and right visual field, brain hemisphere, body. We saw he in the left visual field. We saw art in the right visual field. So art will be communicated to the left brain, and the left brain is the speaking part, so when asked to say which word he saw, the patient will say art, because the left hemisphere, which corresponds to the right visual field, is the hemisphere responsible for language speech. So the patient will say that they saw the word art. When asked to point to which word they saw, which hand will point to which word? So following along, the right hand will point to art, whereas the left hand will point to he. So that is how that works. We will talk about this more in session. If you have any questions, please bring them up in the Zoom session. I went over this really quickly, but again, you can back up the video and re-go through that if it was too quick for you. But that is the end of this worksheet. So make sure to turn in your pre-session worksheet, upload it to Canvas, and then I will see you at the Zoom session.